Hello, I'm Lisa Brouellette, and I'm a master's student at the University of Minnesota Duluth. In this presentation, I will be discussing the work I conducted for my master's thesis on the use of cloacal swabbing to study the diet of raptors during migration using DNA metabarcoding. The objective of my project was to develop a method to study diet in raptors during migration. I'll start off with some general background about why dietary studies are important and how traditional methods of studying raptor diet would be difficult to apply to migrating raptors. Then I'll describe my methods on how I used cloacal swabbing and DNA metabarcoding to study diet. Finally, I'll show you what I found and discuss some of the implications of my work. Let's start off by discussing why dietary studies are important. First, understanding diet is important for answering basic scientific questions about ecology. Most dietary studies for raptors take place during the breeding season, and the breeding season diet may or may not represent a species' diet throughout the whole year. Diet may vary throughout the year due to local seasonal changes in prey abundance, and because some raptors migrate to very different habitats for the winter. In addition to seasonal variation in diet, there also needs to be more research on how diet may vary within a species, such as differences in diet between adults and juveniles and males and females. Understanding the diet of raptors is also important for addressing conservation concerns. For example, dietary studies can be used to inform land management decisions. If you want to provide habitat for raptors, it's important to make sure you're providing the right habitat for their prey. While many natural areas, like Hawk Ridge, may want to ensure they're providing suitable habitat for raptors, other places, like airports, may want to actively deter raptors. The study on the right analyzed stomach contents from birds hit and killed by airplanes so that they could advise airports how to landscape to reduce food availability. Arguably, the greatest conservation concern of our generation is climate change. Northern Minnesota is warming quickly and also seeing an increase in rainfall. We know this change in climate is going to alter our forests in northern Minnesota. Changes in habitat type may change prey availability, and we don't know how this will affect raptors. And so by studying diet, we can try and predict how climate change will affect raptor populations. Another reason why studying diet is important to conservation is because some environmental contaminants biomagnify through the food chain, meaning that species that feed at higher trophic levels have higher exposure risks to environmental contaminants. Next, I'm going to discuss some of the traditional methods that have been used to study the diet of raptors and explain why these methods would be difficult to use during the migration season. One of the earliest methods used to study diet in raptors was stomach content analysis. This was a popular method in the late 19th and early 20th century. It involved going out and shooting a bunch of raptors and cutting them open to see if there was something in their stomachs. Shooting raptors is no longer an acceptable method, so stomach content analysis is really only used nowadays to study the diet of raptors that are already dead, like those that are killed from lead poisoning or hit by airplanes. Another early method of studying diet was pellet analysis. A pellet is just a regurgitated lump of non-digestible material like fur, feathers, and bones. All raptors cough up pellets, but hawk, falcon, and eagle stomachs do a pretty good job at breaking down bone, so their pellets are mostly just fluff, while all pellets have these really nice little intact skeletons in them. So most pellet studies focus on owls. The prey remains in the pellet can be identified by either morphological features or through DNA analysis. And a pellet study would be difficult to do on migrating raptors because you need to have a place to collect the pellets from, like a nest or a favorite roost site, and migrating birds are changing locations constantly, so it would be very difficult to collect enough pellets for a study. Probably the most common method used to study raptor diet is to do some sort of study at a nest. This could involve collecting prey remains or setting up a camera. Both methods have their own limitations and biases, but the biggest problem with studying raptor diet at the nest is that it's highly biased to the breeding season, so you're only getting a brief snapshot out of a few months of the year of what the raptor is eating. And then the final traditional method is direct observation. This is obviously difficult to do for nocturnal species. It's also very time intensive, and precise prey identification is difficult. Those are some of the old methods of studying diet, but now let's talk about DNA metabarcoding. DNA metabarcoding is used to identify species in samples that have DNA from a lot of different species. It uses a unique barcode to identify each species in the sample. This barcode is just a locus or region on the genome that is unique to each species. These barcodes are then sequenced using high-throughput sequencing, which can sequence multiple different DNA sequences at the same time. DNA metabarcoding has been used in a variety of different types of studies. It's been used to study microbiomes. It's been used to identify species for environmental surveys from soil and water samples. And of course, it's been used in dietary studies. So dietary studies that use DNA metabarcoding commonly use feces as the source of prey DNA. As you might expect, it's kind of difficult to obtain raptor feces. It's not like you can just walk around the woods and scoop some up. For songbirds, this is kind of straightforward. All you need to do is catch a songbird and put it in a box for a few minutes and wait for it to poop. But you wouldn't want to try this for a raptor because they are very high-strung birds, and if you try to contain them like that, they could end up injuring themselves trying to escape. So this meant that I needed to come up with a different method of obtaining feces. Let's do a little review about bird anatomy. The cloaca is the orifice that birds use for feces, nitrogenous waste, and reproduction. Swabbing of cloacas has been used for a variety of different research purposes. It's been used to detect diseases like avian flu and West Nile virus. It's been used to study microbiomes and to collect genetic material. Cloacal swabbing has not, to our knowledge, in search of the scientific literature, ever been used to study diet until now. So my project had two main hypotheses. The first hypothesis was just that cloacal swabbing could be used to obtain DNA of food or prey. And the second hypothesis was that the amount of dietary DNA on the cloacal swabs would vary between species of raptors. So for my first hypothesis, I foresaw two possible outcomes. First, that the cloacal swabs would detect dietary DNA, in which case I'd be a super happy master student, or that they would not detect dietary DNA. And I thought of three possible explanations about why the cloacal swabs might not detect dietary DNA. First, that raptors were just simply feeding infrequently during migration. Second, that even if they were feeding frequently during migration, that there would just be a low concentration of fecal residue in the cloaca. And third, that possibly the dietary DNA in the cloaca would just be too degraded to be identified. For my second hypothesis, I thought of two possible ways that raptor species would differ from one another in the amount of dietary DNA detected on their cloacal swabs. So, raptor species differ quite dramatically in their physical size, and I thought that large raptor species, like red-tailed hawks, would have more dietary DNA on the cloacal swabs because they have larger cloacas and I'd be able to obtain more fecal material. And then I thought that smaller raptors, like this northern saw-whet owl, would have less dietary DNA on their cloacal swabs because they have smaller cloacas, and I thought I wouldn't be able to get as much dietary DNA from those feces. Another way that raptor species differ from one another is the way that they fly during migration. I'll just call this mode of migration today. So some raptors, like falcons, migrate with an active flight, by which I mean they have to flap their wings a lot when they're flying. And this is a very energetically costly type of flight. So I expected that these species would need to feed frequently during migration, and that because they were feeding frequently, that their swabs would have more dietary DNA detected. On the other hand, species like red-tailed hawks and eagles primarily soar during migration. I'll call this passive flight. And soaring takes very little energy compared to active flight. So I thought these species wouldn't need to be feeding as frequently, and I expected to see less dietary DNA on the cloacal swabs. So that's it for the background and intro. This is a quick overview about how DNA metabarcoding works. I'll go through each one of these steps briefly. First up is the sample collection. I collected cloacal swabs from migrating raptors in 2019 during spring and fall migration. I collected 71 swabs in late April and early May along the southern shores of Lake Superior. Most of these samples were from sharp-shinned hawks, and they're included in the sample totals in the table on the right. The bulk of my sample collection happened at Hawk Ridge during the fall migration between August and November 2019. I collected samples from 11 different species of raptors. You might notice that the number of samples I collected per species varied greatly. This is just because some species of raptors are caught much more frequently than others, and I just chose to prioritize getting the most swabs from the widest variety of species. So after the samples are collected, then you extract the DNA. After the DNA has been extracted, the next step is to amplify the DNA using polymerase chain reaction, or PCR, which artificially creates copies of the barcode or sequence of DNA we're interested in. The barcode we used was an approximately 171 base pair section of the 12S mitochondrial gene. And we used two different types of primers to amplify our barcodes. We used the MyBird primer, which targeted avian species, and the MyMammal primer, which targeted mammalian species. After the PCR amplification of the DNA barcode, the barcode is sequenced using high-throughput sequencing, which can sequence many different DNA strands at the same time. High-throughput sequencing generates millions of sequence reads, and the bioinformatics step groups the same sequences together into what are called operational taxonomic units, or OTUs. For our purposes today, it's okay to think of OTUs as a proxy for species. That's not an exactly correct definition, and here's why. If we have two closely related species with the same sequence of DNA for our barcode, then they will be grouped together into the same OTU. Likewise, if we have two individuals of the same species and they have unique sequences for the barcode, then they will be grouped into two different OTUs. But we chose our barcode because the locus had been shown to be unique between species with little variation within a species. So for today, it's fine to think of an OTU as synonymous for species. Just know that's not always the case. The final step of DNA metabarcoding is to identify those OTUs by searching a database and seeing if there's a match for the sequence. I used the NCBI's GenBank database. So that's it for my methods. Let's go on now to of the 287 cloacal swabs I analyzed, 18.46% had DNA from at least one prey species. The majority of the swabs with prey DNA detected one prey OTU, 8 swabs detected 2 prey OTUs, and 2 cloacal swabs had 3 or more prey OTUs. Of the 11 species of raptors I swabbed, 5 species had samples with prey detections. I think the main reason I did not see any prey detections for the other 6 raptor species is simply because of a small sample size for those species. The graph here is showing the total number of swabs per species with a prey OTU detected. The colors of the bars show whether the prey on the swab were birds or mammals. The purple means that the swab had 2 or more OTUs that included both bird and mammal DNA. Based on what we know about the diet of these raptor species, the types of prey that I found matched up nicely with each species' expected diet. Sharp-shinned hawks prey primarily on birds. Cooper's hawks and northern goshawks hunt a mixture of birds and mammals, and red-tailed hawks and northern saw-whet owls eat mostly mammals. Let's take a closer look now at the identity of these prey OTUs. There were a total of 35 prey OTUs detected on the Cloico swabs. 19 of those 35 had a perfect or very close match in GenBank, meaning that we could confidently conclude the identity of the OTU. On the right are some photos of the species that we were able to confidently identify to species level. So what about those 16 OTUs without a close match in GenBank? Those are still real prey detections. It just means that whatever species the DNA came from is missing from the database. That means we had to assign those OTUs to a higher taxonomic level. For example, we could say the DNA came from a vole, but we just don't know what species of vole. As for the types of prey found on the swabs, 23 OTUs were from birds and 12 OTUs were from mammals. And I'm not going to have time today to show you all the different prey detections and all the different swabs, but I would like to take a few minutes to highlight some of the interesting things I found. The first case is of a northern goshawk swab that had opossum DNA. I thought this was really interesting because if you're from Duluth, you know that opossums are not very common in our area. In fact, opossums are not even native to Minnesota, but over the past century, their range has slowly moved northwards in the state. I found this case especially intriguing because it made me wonder if this was a northern goshawk that ate an opossum around Duluth or north of here on its migration south, or did this goshawk eat an opossum south of Duluth where opossums are more common and then fly north? Even though the sample was collected in the fall, we'd be expecting raptors to be migrating south. I can't say what happened here, but this case really highlights to me that DNA metabarcoding of dietary samples can be used for more than just describing the diet of a species. These types of samples can be used to detect rare species in an environment, or they could be used to tell us something more about the behavior of a species, such as its migratory movements. On a final note, although northern goshawks are large hawks that would be capable of killing a small opossum, I just want to emphasize that we can't tell if this was a predation or a scavenging event. Northern goshawks have been occasionally documented to scavenge, so it's definitely something to consider. The next interesting case was of several sharp-shinned hawk swabs that contained turkey DNA. These 3 samples were all collected in the spring. Now, if you aren't familiar with sharp-shinned hawks and you're wondering if one could kill an adult turkey, the answer is no. So what's the deal here? I thought of a few possible explanations. First, that they had simply eaten a turkey chick. But wild turkey chicks in Minnesota and Wisconsin don't hatch until early June, and these samples were collected in late April and early May. And free-range turkey farms in our area would not be letting young chicks out of the barn at that time of year because of the cold. Wild turkey chicks do start to hatch in late April in southern Texas and Florida, but it would take several days for a sharp-shinned hawk to fly from the southern U.S. to Duluth. And while we don't know how long prey DNA can hang out in the cloaca for, the average gut retention time for raptors is about 6 to 8 hours. So it doesn't seem likely that a swab would detect prey eaten more than 24 hours before the sample was collected. The most logical explanation I could come up with was that these sharp-shinned hawks had been scavenging turkey remains. Now, turkey hunting season in Minnesota and Wisconsin runs from mid-April to the end of May, and I thought this provided a nice explanation for a source of turkey remains for sharp-shinned hawks to be scavenging. I should note, though, that I could not find any records of sharp-shinned hawk scavenging in the literature, which is really what makes this such an interesting finding. Outside of the migration season, sharp-shinned hawks are secretive little raptors, so I see this as pretty compelling evidence that there needs to be more research done on the feeding habits of sharp-shinned hawks, especially during migration. The next interesting finding I want to discuss is a difference in prey detection between spring and fall samples and the effect I think season has on feeding frequency. So we're just going to look at sharp-shinned hawk data here because it's the only species I sampled in large numbers in the spring and the fall. So I collected 141 total swabs. About 45% of those swabs came from the spring and the other 55% came from the fall. But when we look at the percentage of swabs with prey detections, 27% of the spring samples had prey detections, while only 14% of the fall samples had prey detections for sharp-shinned hawks. So I ran a chi-squared statistical test to see if this difference in detection rate between the spring and the fall was a significant difference. And the chi-squared p-value came back as 0.057. And if you're not familiar with chi-squared tests or p-values, this just means that this difference I found in prey detections between the spring and the fall samples is, maybe borderline significant. And I think it indicates that there may indeed be a difference in feeding frequency between the spring and the fall, and that it justifies more research. The last little thing I'm going to talk about today is how I tested my second hypothesis. This was the hypothesis where I thought that the amount of dietary DNA on a cloacal swab would vary between species based on differences in their physical size and differences in the way that species fly during migration. In order to test the effect of species, size, and mode of migration on the probability of detecting a prey species on a cloacal swab, I built 7 generalized linear models and ranked them by Aic Score. Aic scores are just a statistic that scores a model by how much variation in the data it explains, and it also punishes a model for being too complicated. In the table on the right, the best model was the model that just used species as the predictor variable. This model was much better than the null, and the 3 other models that scored better than the null were really only better because they included species as a covariate. Once species is removed from those models, we see that mode of migration and size are not good predictors of prey DNA detection. Let's take a closer look now at the probability of detecting prey on a cloacal swab based on species. The error bars here are quite large for a few of these species, just because I had a low sample size. The species with the highest probability of prey detections were the northern goshawk and the northern saw-whet owl, and the species with the lowest probability of prey detection is the red-tailed hawk. Now, onto my conclusion. First and most importantly, it worked. Cloacal swabs can be used to study diet, but an 18.46% prey detection rate is pretty low, so I'm going to talk briefly about what I think can be done to improve the method. First and foremost, a controlled study would be the best way to answer questions like how long prey DNA persists in the cloaca, what's the ideal time to swab a cloaca after a raptor has eaten, and which raptor species have the most prey DNA on cloacal swabs. Secondly, there are definitely things that could be done to refine the molecular methods. We use the 12S locus, but maybe there's a better loci that could be used. We could also try using different types of primers for PCR. We know several of these raptor species eat insects, but the primers we chose for PCR did not amplify any arthropod sequences. There are certainly primers out there that could do this, but we were simply limited financially, so we chose to prioritize the avian and mammalian prey. Next, we could try things like taking multiple swabs from a single bird and seeing if that provides more DNA. We could also try things like swabbing other parts of a raptor, like the beak, mouth, or talons, and comparing those to cloacal swabs. More research on the feeding frequency of different species of raptors would be helpful because it would help us to know which species are more likely to have prey detections. Given what I found, I don't think it's worth trying to use this method on a species that's feeding very infrequently during migration. And finally, I think it's important that we try and factor in the time of day into when the cloacal swabs are collected. As I mentioned earlier, the mean gut retention time for raptors is about 6 to 8 hours, so maybe it's better to collect swabs from diurnal species in the afternoon and collect swabs from nocturnal species late at night or into the early morning. To wrap up here, I'll just give some quick pros and cons of cloacal swabbing. First, for the pros, you can use it on any species of raptor. It's relatively quick and easy to take a cloacal swab. Even though that my project was only looking at diet during migration, you could certainly collect cloacal swabs at any time of the year. And there may be fewer biases in cloacal swabbing than other methods of studying diet, but this really depends on the barcodes and primers you use. Plus, we really don't know if there are some types of prey that are more likely to be detected in the cloaca than others. For the cons, the main drawback is that we had a very low return of prey DNA on the cloacal swabs. So if you want to be able to say something about the diet of a species, you have to swab a lot of cloacas. And DNA metabarcoding is expensive. Another drawback is that you need to capture a raptor. Now I want to emphasize no raptors were harmed in the making of this project, but I'm including this as a con because some species of raptors are very difficult to catch, and if you can't catch it, you can't swab it. And finally, you have to say cloacal a lot. That's it for me. Thank you for watching. As with every project, there are many people and organizations to thank. I received funding for this project from the Bell Museum of Natural History's McKinney Fund and Duluth Superior Area Community Foundation's Biodiversity Fund provided a grant to Hawk Ridge Bird Observatory to fund this project. Huge thanks to Hawk Ridge Bird Observatory and their banding director, Frank Nicoletti. This project would not have been possible without them. All the laboratory work for my project occurred at the Environmental Genetics Lab at the US EPA in Cincinnati, Ohio, under the direction of Dr. Aic Score. Thank you to my graduate program, the Integrated Biosciences program at the University of Minnesota Duluth. And finally, I'd like to thank my graduate committee. Dr. Matthew Ederson is my advisor. Dr. Don Brandstrader is faculty in the biology department at UMD. Dr. Julia Ponder is the director of the Raptor Center on the Twin Cities campus of the U of M. And Dr. Aic Score runs the Environmental Genetics Laboratory at the US EPA. And thank you again for watching.